The overall goal of this procedure is to evaluate the NK cell's cytotoxic response to HIV-1 infected cells. This is accomplished by first isolating and infecting CD4 positive T cells from venous blood. Then, natural killer cells are isolated from the same donor. Next, uninfected CD4 positive T cells are isolated away from the infected cells. Finally, the infected cells and the NK cells are used as targets and effectors respectively in one of two assays to assess NK cell cytotoxicity. Ultimately, results can be obtained that show that NK cells can respond to HIV-1 infected cells through upregulation of CD107A expression as determined by flow cytometric analysis or through chromium-51 release as detected by a gamma counter.
This method can answer key questions in the HIV-1 field, such as do NK cells respond to HIV-1 infected cells? What receptor ligand interactions are important in NK T cell interactions? And do HIV-1 viral proteins modulate the NK cell response to HIV-1 infected cells?
After stimulating CD4 positive T cells that have been isolated from human peripheral blood and stimulated by anti-CD3, anti-CD28 coupled beads for 72 hours, count the resulting T cell population by hemocytometer and then split the culture into two groups. Add approximately 2 × 10⁶ T cells to a 15 ml conical tube for use as the uninfected control group and place the remainder of the CD4 positive T cell suspension into a 50 ml conical tube for the infection group. Centrifuge both groups at 300 G at 22 degrees Celsius for 10 minutes and then aspirate the supernatant. Resuspend the CD4 positive T cells from the 50 ml tube directly in culture fluids containing virons and add polybrine to a final concentration.